We're live.
All right. And welcome everyone to Standing For Truth Ministries. We have a good one today. A live debate between Jackson Roe and Standing For Truth himself, Donny. So this one's going to be good. The debate is on, is all life related through common ancestry? And just to let everybody know that there is going to be an after show on Logical Probable Possibles channel after this. He will have a link probably in the side chat. I think I just see it right now. So that'll be good. The debate format is going to be a 17-minute opening with a 10-minute rebuttal each person, with a 35-minute discussion and a 5-minute closing, with followed by audience Q&A. So make sure to either tag myself or Donny or Standing For Truth. We'll see them on the side coming through, and we will gather them up. So don't forget to do that, because it is an indeterminate time that we will try to go and have questions get answered until we burn out, of course. So without further ado, I'm going to have our debaters give an introduction about themselves and why you guys are here, and feel free to take it away. We will pass it over to the channel owner himself. Wham, where are you in this corner?
Thanks for the opening there, Matt, our award-winning moderator. So I appreciate it. Matt was just here last week debating Jackson. I was moderating. Now I'm debating Jackson and Matt's moderating. So I appreciate that. And yeah, my name's Donny, also known as Standing For Truth. Usually I'm the one sitting here moderating. But tonight I am debating one of my favorite topics, which I know this is also Jackson's favorite topic, and I guess his specialty. So I'm looking forward to this. This is my formal debate number 95 if we were to count all of the impromptu debates that take place on people's channel, like Logical, Plausible, Probable, all the impromptu open mic debates, I'm probably at like 200 or something. But I love, these types of sophisticated discussions. Critical thinking is important. I've written numerous books on this topic, so I'm looking forward to this. You guys can check our website, standingfortruthministries.com, where we've got endless content, including, articles, books, videos, lectures, so on and so forth. So, yeah, enough for me. Thanks for doing this, Jackson.
Thanks for having me again. I'm pretty popular on this channel lately. Yeah, anyway, I crossed 400 subscribers, so it's a big deal.
Nice.
I mean, not really, but And kind of unrelated, something that does affect me though. They're passing it doesn't affect you at all, Donny. You're in Canada, but they're passing a bill that effectively bans all exotic animals from traveling interstate or being imported into the country. So anything past a leopard gecko, you're basically not going to be able to get anymore. So it passed the House. It's going to pass the Senate. It's pretty much a disaster. So call your senators if you can. I mean, well, not that ever works, but yeah, that's pretty much all I have to say. I just wanted to say that, just do something.
All right. If you guys are ready, then who is going first on this one? I'll get the timer.
I guess it'll be Jackson, since he's the affirmative tonight.
Okay.
As he would say yes to the debate thesis. So, Jackson, if you want to kick us off, my good man.
Go ahead and share my screen. Let me see.
And whatever time's not used, as we always do, Matt will throw it into the audience Q&A.
All right, can you guys see it?
Yes.
Yep.
Okay. This is the graph that Ken Hovind always uses to make fun of the idea of common ancestry. So I thought I'd just use it just to play into it. But yes, everything is technically related. That doesn't mean everything is closely related. Like the bird and the octopus, they're not very closely related, but they are distantly related by several 100 million years. Sounds crazy on its face, but it's true. Here's a phylogenetic tree of mammals, tracking several genes, and it makes a tree and a phylogeny that you wouldn't expect if they weren't all related somehow. It's just a very big coincidence that God would create something like this, especially created in 6 days, and have it all track like this. It doesn't make sense. Here's the overall tree of life, phylogeny of life: eukaryotes, bacteria, archaea, and they're all linked together, distantly, of course, but they are all linked. Now, here's a funny example. Hippos and whales are the two closest living relatives to each other, and artiodactyls are more distantly related. And whales came from artiodactyl ancestors, that's proven in the fossil record, like Pachycetus, an artiodactyl, but with a skull very much like a whale. And there's a clear progression, like Ambulocetus. Those are one of the ones everyone knows about. And there's Myocetus, which is a fairly recent discovery. And there's Georgocetus, which is basically just Basilosaurus with legs, more developed legs. This is Durodon, a basilosaurid whale. You can see the skulls are very similar throughout the progression. So has some hind limbs, but they're basically useless, definitely useless for walking for their intended purpose. They have heterodont teeth, unlike modern whales, molars, premolars, canines, incisors. They don't have a melon on their head. They have an intermediate blowhole, kind of up halfway up the snout. They have a smaller brain, a lot of primitive features. So this in itself would prove that whales are from land animals. Even without the walking whales, this thing would do it, I think. Skipping right over to something totally unrelated, well, distantly related, of course. That's the argument. But turtles. This is what the turtle came from, a diapsid reptile with broadened ribs like this. Then it progressed gradually to something like odontochelys, which basically had half a shell. It had a plastron but no carapace, had thickened ribs on its back, but no carapace, no true carapace. Skipping again to earlier in the tree, this is Parmastega, which is a tetrapodomorph fish land animal thing. It's the best way to explain that to the layman. This is, of course, Tiktaalik, probably the most famous one on the list. It was predicted to be found exactly where it was found in Devonian strata, exactly where it was found on the place on Earth, Ellesmere Island, in the Arctic Circle. They went to the Arctic Circle expecting to find something like this, and lo and behold, they did, which is a pretty big coincidence. This is Elpistostegi, which is basically Tiktaalik 2.0. It has the beginnings of digits in its fins. This is Acanthostega, the next iteration. There's a little bit of a gap between the two. It's yet to be found. But this is Acanthostega. It has internal gills and fin rays and 8 digits. It's still technically a fish, but it looks very much like a tetrapod. This is Ichthyostega. In the center is the actual skeleton and a diagram or a CT scan. I can't really tell. But you can see this is still technically a fish with internal gills and tail fin rays. So very strange shaped fish. But it's got 7 digits, 3 digits forming into 1 digit. So a transitional structure. Then going back in time again, I don't have these in order. This is Bicellum brazeri, about 1 billion years old. The first known multicellular organism. It has 2 types of differentiated cells. Then we skip ahead a little bit to the Cambrian, one of the first chordates, Hycuictis. And the Silurian, one of the first jawed fish, maxillate placoderm, had premaxilla, maxilla, and a dentary bone, the first fish to have that feature, the first modern jaw. There are 4 species of those that are known. Now skipping way ahead, again, they're out of order. This is Australopithecus prometheus, the Littlefoot specimen. This was found almost completely articulated in Stertfontein Cave, South Africa. It was encased in limestone. It took years to fully drill it out. But you can tell from the femur structure, the hip structure, the spine structure, the rib structure, the knee structure, the ankle bone. Everything about it says that it walked upright. This is the big man fossil, which is not very complete, but it's complete enough. It's an Australopithecus afarensis, the same as Lucy. Only this one is 5 feet tall. Again, has the knee, which creationists argue about Lucy's knee a lot. Well, problem solved. We found another one. And another one, in fact, on this one, Australopithecus afarensis. This is a juvenile specimen. Skipping back in time again. This is the mantle progression. In the early Permian, you had pelycosaur synapsids like these. This is Dimetrodon. Then you skip ahead to therapsids. In the late Permian, this is a Gorgonopsian therapsid. I forget what it's called. This is a cynodont of the mostly Triassic. More mammal-like reptile, but still a reptile. I already talked about this, but this is kind of limb evolution. This is Elpistostega. You see digits forming here. Acanthostega with the 8 digits. And Ichthyostega with the 7 digits. Now this one's one of my favorite lines, the theropod bird line. This is Anchiornis. I actually have a canvas of this one behind me. You'll see in a minute. 160 million years old. Here's what it actually looked like. Actually, I can tell the color of the feathers from fossilized melanosomes in the fossil. This is Eosinopteryx, another 160 million year old one, predates Archaeopteryx by 10 million years. Kai Hong, actually older at 161 million years. Aornis, 160 million. This is Protopteryx, about 130 million. years old. Now, clearly at this point, it's more bird than anything else, but it still has 3 infused digits, teeth, and its feathers are much more primitive in structure. Another way you can tell things are all related to each other: chickens can still produce teeth. Dolphins can still produce hind limbs, atavisms. Here's something about more about whale evolution. They can tell those walking whales are whales because of the very diagnostic ear structure, not shared with anything else on the planet. This is an Indo-Hyas skull next to a raccoon skull. On the surface, they look kind of similar, but the teeth and, of course, the auditory bulla give it away. This in the middle is a pronghorn. Next to the walking whales, I'll just say walking whales. I don't want to say their names. It's too complicated. But they are clearly artiodactyls based on their ankle joints. Here's Basilosaurus, which everyone agrees is a whale, even big creationist groups like Ancestry and Genesis, but still has the same kind of ankle joints, kind of fused though, down to 3 digits each. So it's kind of wasting away as time goes on. This is a seal inner ear structure. I bring this up because I saw a talk or read an article once from David Minton, which I believe he recently died, and I don't want to speak ill of the dead. He seemed like a nice guy. But he was very wrong when he said that seals and these walking whales are related. He said they were pinnipeds, and that just doesn't make any sense. This is about single-celled algae. When introduced a paramecium, a predator, 50 weeks, 700 generations, 2 of 5 experimental populations developed multicellular life, a multicellular kind of colony. And they weren't just colonies, they formed a cell membrane, and they reproduced by kind of budding, I guess is the best way to put it, instead of cell division like these would in the single celled. So we kind of saw multicellular life evolve independently. And they stayed that way for 4 years. They ran the experiment that long. Here's a manatee ancestor, Pasosiren, a quadruped. And there's a line of those like the whales. They're just harder to find photos of. So here's a diagram of them. Here's some basic genetic evidence. I'll just read the titles for the sake of time. Reconstructing the genome of the most recent common ancestor of flowering plants. Last common ancestor of green algae and land plants. Ancestor of animals, fungi. Determining the divergence time of the major kingdoms of living organisms with a protein clock. Primate molecular divergence dates. So this stuff is well known. I won't really go into it here because I'm not going to read all that and waste time. But yeah. One last thing. I got a bunch of stuff for rebuttals later, hopefully. I think that's where we're really going to get into it. This is just kind of breaking the ice. If everything was specially created, we would see weird animals like this that don't fit anywhere, genetically or anatomically. Like, what is this thing? Is it an amphibian or a bird or a mammal? I don't know. What's this? Or that? Or this thing? They wouldn't fit anywhere. But everything on Earth does fit in the tree, so to speak. And let's see what else I got. I think that's good for now. I'll end it by showing you this. It's a fossil whale vertebra I have. It's 5 million years old, so it's still technically an anatomically modern whale, but it's kind of cool. Thought I would share that. All right.
All right. Good one. You went 12 minutes. So we will add those extra to the Q&A or debate part. All right. And whenever you are ready, let me know and I will start the clock. And you are muted. Don't forget.
Yeah, just getting everything ready here. Let me see. And I'll be screen sharing in 1 second. We've got 17 minutes. So let me share a screen here real quick. And okay, let me start my timer. I appreciate that opening, Jackson. This is going to be fun. Okay, here we go. 17 minutes. Thank you for doing this debate, Jackson. I have written, as I pointed out, numerous books on this topic and look forward to a profitable discussion on it specifically. Here are some of the books I have written. So tonight we are debating the question, is all life related through common ancestry? Jackson here tonight has the burden of proof, as he is in the affirmative. He must provide convincing evidence that not only dogs and wolves are related, but that dogs, wolves, banana plants, and whales are related through common ancestry. Does Jackson believe he is related to a mosquito and a strawberry? The answer is yes, as he demonstrated in his opening statement there. And it is his burden tonight to demonstrate this. Agnostic lines of evidence will not help Jackson win this debate. He spent his entire opening going over agnostic or non-discriminatory lines of evidence. What I mean by that is the sky is blue and the earth is round. Okay, that is not evidence for either creation or evolution, as in that can't help us differentiate between the models, since both evolution and creation can explain why the sky is blue and the earth is round. Jackson must present differentiating lines of evidence that can distinguish between both universal ancestry and separate ancestry. Jackson must explain how universal common ancestry and its proposed mechanisms can account for the four-dimensional code. The genome is not what evolution expected. The examples of mutations we have are not the types required for evolution to advance. Evolution has to explain how the four-dimensional genome with multiple overlapping codes and chock full of meta-information came about. Genetic degeneration is a fatal problem to Jackson's position tonight. What is genetic entropy? It is the genetic degeneration of living things. Genetic entropy is the systematic breakdown of the internal biological information systems that make life alive. Genetic entropy results from genetic mutations, which are typographical errors in the programming of life's instruction manuals. Essentially, evolutionists like Jackson look to mutations as being the source of all genetic variation that natural selection can act upon. Unfortunately for Jackson, natural selection is very limited. It is a fine-tuning mechanism that keeps a species as strong as it can be. It doesn't create anything new. It only selects from what is already there. And mutations, the so-called source of new genetic variation, systematically erode the information that encodes life's many, many essential functions. The human mutation rate is roughly 100 new mutations per person per generation. The 8 mutation rate is similar. Almost none of these mutations are beneficial. The new ENCODE findings reveal that most of the human genome is functional. How does Jackson address this? I'd like him to address this in his rebuttal. Hence, most random changes in the genome must be deleterious. Even if 90% of the genome is perfectly neutral junk, which is no longer feasible, there would still be about 10 harmful mutations arising in every person, every generation. Even the most fit individuals are still more mutant than their parents. So even intense selection against the less fit cannot stop mutation accumulation, because most deleterious mutations cannot be selected away. They will accumulate continuously from one generation to the next. Now, we understand genetic entropy on a personal level, but since mutations are passed on from generation to generation, there is also genetic entropy on a population level. Why can't natural selection filter out all the bad mutations? There are two reasons. There are too many bad mutations continuously pouring into the population. And B, most bad mutations are too subtle to be selectively removed. Epigenetic mutations will consistently magnify this problem. We also have polyconstrained DNA, the fact that most DNA sequences are polyfunctional and so must also be polyconstrained. This means that DNA sequences have meaning on several different levels, and each level of meaning limits possible future change. For example, imagine a sentence which has a very specific message in its normal form, but with an equally coherent message when read backwards. Now, let's suppose that it also has a third message when reading every other letter, and a fourth message when a simple encryption program is used to translate it. Such a message would be polyfunctional and polyconstrained. We know that misspellings in a normal sentence will not normally improve the message, but at least this would be possible. However, a polyconstrained message is fascinating in that it cannot realistically be improved. It can only degenerate. We also know that the evolutionists look to beneficial mutations to supposedly counterbalance the damage done by the accumulating genetic load. Unfortunately, most mutations are reductive. They are rare. And as you can see here, most so-called beneficial mutations that they can point to are degrading to genetic functionality. That is a problem for Jackson here, unfortunately. The arguments that have been put forth against genetic entropy, I'd be curious to see what arguments Jackson here looks to have been systematically analyzed. For example, here on the slide, we've got junk DNA, mutation count mechanism, truncation selection, synergistic epistasis. They've all been looked at and they've all been falsified. As you can see here, the human germline mutation rate, 100 de novo mutations, our future genetic load. Human mutation rate revealed, and you can look at these paper yourselves. Right here, slightly deleterious mutations. This isn't young Earth creationists making this up. Okay, they you see in the literature from population geneticists, very slightly deleterious mutations. We are degenerating every single generation. It seems unlikely that any mutation is truly neutral in the sense that it has no effect on fitness. All mutations must have some effect on fitness. Nearly neutral mutations, again, it seems unlikely that any mutation is truly neutral. The question is, these mutations are unselectable. They're unselectable. So what type of selection can Jackson here present us today that can stop the degeneration process? Because selection can't see most mutations. We know mutations degrade genetic functionality and information content, and mutations are primarily why we die. And as we pointed out earlier, there is genetic degeneration on a population level. Now, what's interesting is Jackson's opening statement is he spent a lot of time on what's called reconstruction in phylogenetic systematics. Okay, and a lot of this involves imagination. Most of Jackson's arguments involve just that. For example, fossil reconstructions and interpretations that Jackson looked to in his opening statement, and these so-called transitional forms in the fossil record, quote unquote, universal common descent is void of an adequate mechanism. As we have seen, most mutations are invisible to selection. They build up and degenerate. Natural selection is limited. It doesn't create anything new. And mutations in natural selection are simply not up to the task to take a single-celled like ancestor into a whale over billions of years. Without an adequate mechanism, any historical reconstruction lacks explanatory power and is therefore not to be viewed as a correct reconstruction. Jackson's reconstruction interpretation of the data, as seen in his opening statement here, including the fossil record data that he spent a great deal of time on, is incorrect. Jackson assumes universal common descent, and because of this presupposition, he imposes it onto the genetic data. This is not a conclusion drawn from the genetic data. Remember, genetics is the best way to answer the question of ancestry. Okay, genes and traits are what is inherited, sperm and egg. Not a fossil, which Jackson looked to, not a bone, not a reconstruction, not a picture. We saw a lot of pictures in his opening statement, not geography, no, genes and traits. And genetics has refuted universal ancestry. Genetics has actually confirmed separate ancestry, and we will get into this a little bit more later. Okay, let's get into some of the most common lines of evidence for universal ancestry and demonstrate why they are not good evidence for fish-to-fisherman evolution, or specifically why they don't help Jackson meet his burden of proof tonight. Notice on the slide here, we have transitional forms that Jackson spent some time looking at, homology, and nested hierarchies. Okay, we have nested hierarchies here. And let's examine each one here. Homology. The biological phenomena that we've explored parallel the properties of family trees, but they also have strong parallels to another concept. In other words, a competing explanation exists for all the evidence we've just discussed. This competing explanation becomes clear with an example. Okay, so we know humans design in homologous patterns. Okay, take the sedan as an example. Whether sedans are manufactured by automakers based in Asia, North America, or Europe, all share a common set of features. You won't find cars without wheels, engines, doors, or windshields. Despite the geographic distance separating their locations, sedans made by Honda, Chevrolet, and Mercedes have four wheels, doors on the side, windshields in the front, brake lights in the back, and a host of other shared characteristics. Now, the reason for this similarity is not common ancestry. Honda, Chevys, and Mercedes did not evolve from a common sedan ancestor in the Arctic. Rather, because these cars were designed for similar functions, these purposes put constraints on the final product. We can go on and on about homology. It's exactly what we'd expect given the design perspective. Same thing with shared embryological patterns. We could extend this logic to the manufacturing process where we would see similarities in blueprints. In fact, it would be not until the end stages of these modes of transportation that we see the largest differences. Okay, same thing with nested hierarchical patterns. Under the young Earth creation model, God designed the entire universe, including the various kinds of biological life that exist in it. And we would expect to find that life fits a design pattern. Since humans are made in God's image, we can get a sense for what kinds of design patterns God may have used by examining the patterns that result from human designs. For example, design means of transportation easily fit a relative hierarchical pattern. This fact is unequivocal. Sedans resemble SUVs more than they resemble tractor trailers, and all three vehicles have more in common than do sedans and amphibious assault vehicles. The latter two vehicles have more in common with one another than with submarines, and this simple pattern matches the type of hierarchy that we see in biology. Again, relative hierarchies are characteristics of design. Of course, we would expect more similarity in genetics, morphology, anatomy, and physiology between a human and a chimpanzee than between a human and a dog, or between a human and a banana. It's exactly what we'd expect. Transitional fossils that he looked to. Okay, these parallels between design and biology extend even further. In our discussion of the deeper fossil layers, which Jackson spent some time on, we observed the presence of fossils that seem to blend the features of two different categories of creatures. Some products of human design also seem to span two categories. As we've observed, boats and tanks would fall into entirely different classes. Yet vehicles exist that blend the features of these two very different types of transportation. For example, amphibious vehicles resist classification. They're the perfect example of a design transitional form, designed for the transitional environment between water and land. In fact, if you were following my classification scheme for vehicles, you might have been mentally protesting. This is a source from Dr. Nathaniel Jensen, where he points out that we have an increasing number of hard to classify vehicles. For example, crossover vehicles, where the distinction between cars and SUVs has become blurred. What is currently labeled a car and labeled an SUV is now a matter of debate. These are designed. Okay, these did not evolve. These were designed for a purpose. We see the Jeep Gladiator here, the Dodge Journey, the Ford Flex, an amphibious assault vehicle. To the evolutionists, this is the perfect example of a so-called transitional form. Notice the slide. We have a unicycle, a bicycle, a tricycle, and a four-wheeler. I could predict, because Jackson talked about predictions with Tiktaalik and so on and so forth, I could predict that since we have unicycles and four-wheelers, there must be evidence for a tricycle and a bicycle out there. And it just so turns out that's exactly what we find. This is not evidence that the unicycle evolved into the four-wheeler. Rather, this is evidence of design. Each design is suited for what it was meant to do. Vestigial organs and structures overturned. What we see is function, function, function. The chromosome 2 fusion has been completely overturned through genetic related data. There's a huge problem that I want Jackson here to address, because we have these very large satellite sequences at the ends of the chromosomes of chimpanzees and apes. Okay, but what we find is that these large ape-specific satellite sequences that should be seen flanking the reputed fusion site on both sides, they are not there. They are not there. They are absent. This is a very serious problem that I would like Jackson to address if there really was a chromosome 2 fusion related to human evolution. And you can find more about this in the Ventura paper. So as we can see here, pseudogenes overturn the so-called genetic identity of human to chimpanzee. Actually reflects, when you consider all genetic regions, only about 88% similarity, which is 400 million DNA differences. There's a huge waiting time problem to fixate that many DNA differences. In my last couple minutes here, I'm just going to cover a few lines of evidence that suggest separate ancestry. Even though the burden of proof today is on Jackson to demonstrate that whales and banana plants are related through common ancestry, agnostic lines of evidence are not going to do it. But there are differentiating lines of evidence that actually support the separate ancestry model. For example, we have a genetic boundary study that was done showing clear genetic boundaries in the CO1 gene through these barcoding studies, and they demonstrated that over 90% of all species arose at the exact same time. The only historical record we have of a recent sudden origin of the diversity of life with distinct boundaries is in the opening chapters of the book of Genesis. The Bible's account is vindicated by science once again. We have the Y chromosome data, where we know the Y chromosome is mutating incredibly fast. Dr. Nathaniel Jensen has discovered that the Y chromosome really only goes back 4,500 years to Y chromosomal Noah, and there's been subsequent papers going over the predictions that are derived from that model. Mitochondrial Eve, the mother of us all, we know that the mitochondrial DNA also mutates fast. There's incredibly low variation. And if we look at a phylogenetic tree, for example, here we see that perfect starburst pattern. Explosive growth from a center point. We only see a few mutations, low variation. We see in some of these haplogroups, for example, HVR, we have some people picking up more mutations than their cousin in the same amount of time that destroys the evolutionary molecular clock. This is exactly what we'd expect given the separate ancestry model. How does Jackson address this? How does Jackson address the evidence from orphan genes, these taxonomically restricted genes that are exactly what the separate ancestry model would suggest? Time flies by. I've only got 30 seconds left here, but we could go on and on and on about the evidence for separate ancestry. This is a debate on universal ancestry, so the burden of proof is on Jackson. But I can tell you, we could spend hours and hours going over the amazing lines of evidence that confirm separate ancestry, and there's 17 minutes, so I will call it quits there. Thank you so much.
All right, Jackson, if you wanted to share screen, go ahead, and I will start whenever you are ready.
I'll share screen here in a minute. I got some notes first. First, you said mutations can't make new information. Well, there's duplication mutations, which can work with existing stuff, copy it, and then it changes through further mutations. Then there's de novo mutations, which I know you express you don't believe in those, but we can get into that later. I've got some notes on that. Then there's horizontal gene transfer from one organism to another, which is not novel in itself, but it's novel to that new organism that it's getting it from. Now, genetic entropy. I've done a lot of work on genetic entropy recently. I'm doing some calculations. I'm trying to equate, for example, let me find it. I got a ton of notes. Like influenza B, it's existed for 4,000 years. It has 14,500 nucleotides, one mutation per cycle. Sometimes some sources say 2, but I said 1 for the sake of argument. It replicates every 6 hours. So you take the amount of human nucleotides, divide that by 14,500, divide that by 100 human mutations, comes out to 4,000 something. 4,000 years times 365, that comes to 1.4 million times 4, because 6 goes into 24 4 times. Comes out to 5.8 million. Multiply 5.8 million times 1 times 4,000, comes out to 23 billion. What am I talking about? Some people are probably lost. I was explaining this to my family, and they didn't know what I was talking about. But I'm equating mutation rates of viruses with the mutation rate of humans and trying to see when genetic entropy takes hold. According to this, it would take humans 23 billion years to suffer some sort of genetic entropy. Viruses mutate very fast, replicate very fast, and they don't have the benefit of recombination that sexual organisms do. Now, we can get more into that later. Let's see, functional genome. See, you actually, a long time ago, I actually wrote this down somewhere. The functional genome, you said in the side chat of one thing, we were having sort of a little debate, Donnie. You said evolution should be selecting out non-functional DNA, and that would disprove evolution. Then you said functional DNA proves that evolution is not real. So it's kind of a contradiction. You can't have your cake and eat it too there. A functional genome, I don't see how it would disprove evolution. I don't see how a totally not, 90% non-functional genome would support it necessarily either. Now, John Sanford, he's worked on this mutation rate. He's equated, he said H1N1 went extinct from slow error catastrophe, he said in his paper. He didn't use the word genetic entropy, but that doesn't really make any sense. Like I said, the influenza B has existed for 4,000 years and has a fairly rapid mutation rate, and it hasn't suffered any ill effects. And a question I have about genetic entropy, which Donnie can answer at some point: at what mutation rate over what period of time with what genome size will result in extinction by genetic entropy? No one can answer that question as far as I can tell. Now I will share my screen while I exit out of it. Hang on a second. Give me a minute. Sorry. You know what? I'll just tell you for now. I will pull it up, but save time. Georgia Purdom said in a slideshow, she had a slide that said there are no beneficial mutations. And below that, she said there are beneficial outcomes to mutations in certain environments, which is just saying there are beneficial mutations, because a beneficial mutation is not going to be beneficial everywhere. A beneficial mutation that lets you breathe oxygen isn't going to be helpful if you're in an atmosphere made of methane. Slightly deleterious mutations, I don't think they're much of a problem. Neutral mutations or near neutral mutations are needed for creating diversity in the genome. Can Jackson provide his calculations for critique? Someone says, yeah, I can later. Now, the walking whales I gave an example of, they're not just tricycles and bikes. They're like, how does it have a whale's ear and an artiodactyl's ankle? There's not really a great explanation for that. You can say, and did you find the unicycle and the strata below the bike, and then find the four-wheeler above the bike and the strata, and radiometrically date all those? I don't think so. Chromosome 2, you mentioned, I think something about probably the telomeres being too short. The telomeres fusion is drastically reduced to like a few hundred copies, which is usually much greater than that. I forget the number. But yeah, there's still obviously telomeres. It's still obviously a fusion. There's a degenerated centromere as well. You said 90% of all life on Earth arose at once. There's a problem with that. It doesn't really say they arose at once. It says they suffered some kind of genetic bottleneck at once, about 100,000 to 200,000 years ago, according to genetics. So that doesn't really fit in the creationist model. Chimpanzee Y chromosome and human Y chromosome. Y chromosome is at the forefront of reproduction, so it's going to go through more selection, especially with the Pan genus, the chimpanzees. They multiple males will mate with a female, so the most virile male has the most dominant sperm will pass on his genes. That's kind of how that works. Now, Jensen, you mentioned Jensen. One of his papers, I'm not sure if it was the one you brought up, but he used mitochondrial DNA and nuclear DNA, I think it was. I may be wrong on that, to set a new kind of creationist molecular clock, but it was contingent on it either being mtDNA or nuclear DNA. And it turns out it was the wrong one, so it nullified his whole paper. Now, let's see. The mitochondrial DNA mutation rate, Jeanson also estimated, and it's totally different from any secular literature or any literature on the mitochondrial mutation rate ever done. It's 35 times faster than what everyone else says that the mitochondrial mutation rate is. The mitochondrial mutation rate is like 3,600 years for one mutation. And he said it's 35 times faster. I forget how I came to that conclusion, but it was a limited study, a small sample size or something like that. And he uses an unrooted tree. So a phylogenetic tree will be like a three-dimensional kind of graph. He uses it flat. He shows relatedness, but he doesn't show ancestry. So it's kind of a misleading graph. I don't know if he does that on purpose or not. I don't think he does. And the last thing I've written down is orphan genes. Like there are different genes that aren't shared between us and chimpanzees. Well, there are fruit. We talked about this before too, in another. We kind of had this debate already, Donnie. But there are orphan genes in different species and even different populations of fruit flies. Now, I think we would all say that fruit flies are related to other fruit flies. So orphan genes aren't really a good line of evidence to disprove common ancestry. That's all I have written down. We can get more into it later, obviously, but yeah, I'll hand it over to Donnie.
All right, pretty good. 9 minutes.
Okay.
All right, let me get my timer going here. Time flies by in these openings and rebuttals, don't they, Jackson?
Yeah, they do.
Quick sip of water. All right. I've got 10 minutes on the clock starting right now. And I've got quite a bit of notes based on everything you said, Jackson, and we can do some serious discussion as well in the discussion portion. So Jackson spent a lot of time on phylogenetics. I did address a lot of this in my opening statement, but the reason why everything is connected is because life exists on the same planet. We are made of the same DNA, RNA, proteins. We breathe oxygen. We eat similar foods. Therefore, similarities in DNA, morphology, anatomy, and physiology is expected by the creation model. Does Jackson propose God should have used different building blocks for each form of life? DNA, RNA, and proteins for some, and then not for others? This argument doesn't really make any sense. And it's also a misrepresentation of the creation model that suggests God created us in his image. Therefore, there should be something about us that reflects the divine. And we've predicted that humans will design things in a way that can give us an idea or sense into how God designed things. And it just so turns out that humans build in homologous patterns. They build so-called transitional forms, right? These nice, interesting mosaics. And they also build in nested hierarchical patterns. Jackson talked about whales. He talked a lot about so-called transitional forms and fossils. He looked to the fish to tetrapod series. Tiktaalik stated that evolution has made fulfilled predictions given the fossil record. Well, Tiktaalik failed miserably because they found tetrapod foot tracks or footprints earlier than Tiktaalik in Poland. Okay, so Tiktaalik wasn't transitioning into anything. We see significant overlap. We see geographical mismatches. And again, Jackson is presupposing that evolution happened. And so this is a conclusion or interpretation that's forced on the genetic data, which is the direct lines of evidence. Okay, his interpretations are wrong given the direct lines of evidence. He talked about the human evolution related candidates, the hominids. He discussed Afarensis, Prometheus, and others. He also discussed the mammal-like reptiles. And he looked to a lot of specific features in morphology and anatomy, such as ankle joints, ear structure, and so on. The problem with Jackson's opening is that we don't have the genetics for most or even all of his examples. Today, we know that there oftentimes exists more variation within the same species than across species. This means without genetics and breeding tests, we can't always tell the difference between homology and convergent evolution. I want to ask Jackson that question. If you have a bone found in the dirt and you don't have any genetics or breeding tests, how can you tell the difference between homology and convergent evolution? Anytime convergent evolution is invoked, it is an admittance by the evolutionist to the lack of uniqueness in the universal phylogenetic tree. So that is a problem. And that's why genetics is the best way to determine ancestry. Not a bone, not a fossil. A fossil is not inherited sperm and egg. If every single dog type today was buried in a flood and dug up thousands of years from now, without the genetics and breeding tests, we could make all sorts of erroneous interpretations or fanciful stories that may or not be true. Okay. The problem with Jackson's understanding of genetic entropy is the fact that, because he said that these neutral mutations build up and they act as a hidden reservoir, essentially, of genetic diversity that natural selection can eventually act upon for future novelty in terms of large-scale evolution. But here's the thing. These neutral mutations are not actually neutral. They're effectively neutral. I showed a number of quotes demonstrating that these mutations are only slightly deleterious. It's like a one single spelling mistake in a book, let's say the size of an encyclopedia, okay, or rust on a car. These build up. On their own, they're inconsequential, but over time, the individual mistakes accumulate, just like in a book or on a car with rust marks. In the genome, these individual mutations build up, they degenerate, and the message is eventually lost. Okay, it's not the individual mistakes, it's the accumulation of these individual mistakes. So Jackson wants to assert that the accumulation of these individual mistakes will eventually be called upon or acted upon for evolutionary advantage, when in fact the buildup of these mistakes actually degenerate. Okay, it's going the wrong way. The change is downhill, it's not uphill. We know that selection involves the death of individuals, so selection is extremely limited. His mechanisms are limited. He doesn't have a mechanism. Therefore, don't tell me that it happened if you can't tell me how it happened. You can't select away an entire population, since that would result in immediate extinction. Selection can only ever slow down the degeneration process. It can never solve the degeneration problem. And I like the way Dr. Robert Carter has pointed out that if our species can't persist for millions of years into the future based on mutation accumulation, which puts shelf lives on genomes, that means our species has not persisted for millions of years into the past. Okay, as a matter of fact, if we look at humans in the fossil record, since Jackson likes to look to the fossil record, we see no evidence of hundreds of thousands of years of human existence. Okay, given the evolutionary model, we can assume that there were about 1 million Homo erectus living across Eurasia, Africa. I mean, they handled a massive distribution. Okay, if we look at their average generation time, let's say it was about 20 or 30 years, that means about every 20 years, you have another 1 million dead bodies. And so after 1 million years, we have over 20 billion dead people. This is 20 billion people, 20 billion Homo erectus, of which we have no evidence ever existed. Okay, we know that Erectus was a sailor. They were a sophisticated people group. They were not primitive, so therefore they would have had campsites, villages, fire. We should have a ton of evidence for their existence, and yet we do not. In my last 4 minutes here, I do want to share a screen real quick because I want to address some of his specific examples. And let's see here. I'm sweating. We got a lot of points to address. So the Australopithecines that he focused on, okay? Australopithecus literally means southern ape. As the name suggests, the Australopiths have the overall body plan of an ordinary ape. Their anatomy indicates they were best suited for life in the trees. It's not surprising that they should share most of their features with living African apes today. They're around 3 to 4 feet tall with long arms and short legs, ape hands and feet with long curved fingers and toes, opposable thumb-like toes, ape size and ape-shaped skulls, funnel-shaped rib cages with high-placed shoulders for suspensory behavior. They also exhibited their own unique morphology, which is what we'd expect in the pre-flood world and the immediate post-flood world. Okay, the Australopithecines that are found are typically post-flood. Here's the thing. In this post-flood world today, this is only a snapshot of the overall diversity that ever existed on the planet. So we're only getting a snapshot. Look at how many rhino species today exist. 5. Look at the fossil record. We've got a massive variation of just the rhino type in general. I mean, rhinos with huge horns, middle-sized horns, no horns, small horns. There is a greater amount of diversity in the pre-flood world. So what we see in terms of Jackson's examples here are just examples of pre or post-flood genetic diversity. And he wants to line it up any way he wants. With that amount of diversity, you can make any story. You can make any story from that. We can get into the ordering of the fossils a little bit later if he wants to, but I do want to touch a little bit on his other examples in the last 2 minutes. So the therapsids, Mammal-like reptiles, for example, I had a paleontologist on, Dr. Matthew McLean, and he did a presentation showing discontinuities rather than continuities in the therapsids as well as Dinosauria. So we actually see genetic boundaries and boundaries in the fossil record, including created kinds of the therapsids. We see that evolutionists use selective data using the fossil record selectively. I talked about geographical mismatches that we find in the fossil record as well. Another good point, similarity does not equate relationship. I need to know from the fossil record, okay, how can Jackson differentiate between homology and also convergent evolution? And we have example after example of convergent evolution here that essentially is just a rescue device. Okay, we could go into all the whale evolution examples. Pakicetus, there's been a drastic revision with Pakicetus, Ambulocetus as well. We know that the so-called vestigial hind legs are utilized and functional for reproductive purposes. Okay, the presumed transitional forms, including Pakicetus, Ambulocetus, and Rhodocetus, are highly questionable. The claim that these are all whale precursors is based more on speculation and wishful thinking. Remember, a lot of this fossil record data, guys, is highly fragmented. Again, we don't have the genetics and the complex adaptations that would be required to convert a land mammal to a fully aquatic whale are unimaginable. Even according to the evolutionist time frame, there's a huge waiting time problem in terms of fixating just means to get stuck in place. These amount of beneficial mutations for this type of evolutionary transition. I've got water to land refutations as well. Tiktaalik, again, failed prediction, but unfortunately I'm out of time. So why don't we end it there and then we can get into a little bit more detail.
Awesome, man. You were on the timer better than me. That's good.
1 second to spare.
No problem. And just a reminder to the audience while you get ready is that I would like to tell everybody to tag me if you can, or just throw a question in the side chat and I will compile them for everybody. And Standing for Truth, if you want to play a clip for a commercial, let everybody know. It's okay if not. You're probably already gone. But what's going on is we have a lot of debates coming up. Oh, thank you for
drank too much coffee. That's what's going on.
All right. Here we got some questions coming in already. So I'm posting them in the side for you as they come. This is great. Thank you, everyone. These questions are I didn't have any yet, so this is really good. All right. Here we go. Thank you, everyone. And we have Donny working on a couple things right now. One of the things that's keeping him busy is the production of a new book coming out. And as you would imagine, trying to do debates and write at the same time is really hard. So he will be quite constrained. I don't know. I think he's taking debates, but another question for Jackson. Here we go. From SpongeBob himself. Thank you, SpongeBob. Love the name, too. And thanks, George Bond. I got your question, and I got the question above that.
It looks like, Jackson, you won in terms of bladder endurance. So you took an hour. I had to go for 20 seconds. You win, at least in terms of that battle.
Yeah, I won the bladder battle, that's for sure.
And now we're good for 35 minutes. So whatever you want to talk about, we got a lot of points here to discuss. So
yeah, I got notes. You said this is the open discussion, right?
Yes.
Okay.
Open discussion now.
Therapsid kinds. Could you be more specific about what that means?
Sure. Yeah. If I screen share here, okay. So we have baraminologists, for example, Dr. Todd Wood, Dr. Kurt Wise, Dr. Matthew McLean are a few that come to mind, and they have done studies and analyses using fossil data, morphology, anatomy. And what they do is they take a number of traits and characteristics and they look for continuities. or discontinuities. Now, the created kind would reflect a continuous, right, where you have a group that has continuity within it, but outside of it, you have discontinuity. And that's what they've discovered, specifically Dr. Matthew McLean, when it comes to the therapsids, that there's enough discontinuity in the fossil examples or specimens that we can safely say that there were distinct created kinds of the therapsids. They've also done studies on Dinosauria and the in humans, where we have, this is the last thing I'll say, where we have the separate ancestry model of humans, the Homo group, ape-like group, or the Australopithecines, and then the artificial species wastebasket taxon. So this all comes down to, you know, the phylogeny challenge, or where can we separate ancestry. We've done just that in a number of groups, including humans, where we know that for the most part, we can categorize the fossil examples as either fully human, fully ape, degenerate ape, or wastebasket taxon, as I believe we see with some of the afarensis examples, but especially with habilis and sediba. Go ahead, Jackson.
All right. You're talking like what is it called? Dinocephalian and Gorgonopsian therapsids, like those are different kinds, maybe.
Yeah.
Something
You
like
can
that.
you can read the examples or watch the presentation, but
Yeah, I haven't seen that one.
here's a couple of the papers here that you can, you know, they're technical and long. But this one is specifically with the Dinosauria, baraminological and multivariate patterns, the proceedings of the International Conference of Creation. And he goes into the amount of characters and taxa and characteristics that they looked into. And they go through just how many possible created kinds there are in these fossil groups. But let me kind of turn this around on you, because even baraminology, when it comes to the fossil data, I think is also highly flawed, as in it's restricted. Again, it's genetics, genes, traits, and genetics is what's inherited sperm and egg, not a fossil, not a bone, not geography. So my question to you is, and I think this is the problem we also face when it comes to baraminology in just strictly fossil data. How can we tell the difference if we don't have genetics or breeding tests, Jackson? How can we tell the difference between homology or convergent evolution? Because today we know there oftentimes exists more variation or more diversity within the same species than across species. We see that with Canis domesticus, Canis lupus, Canis coyote, so on and so forth. So how can you differentiate that without genetics and breeding
Right.
tests?
Like, let's take, for example, the platypus bill and the duck bill. On the surface, they look kind of similar, you know? Would you agree with that?
Yes.
Yeah. Well, when you get down to it, they're not very similar. The platypus bill is electroreceptive. The duck bill isn't. The platypus bill is leathery. The duck bill is solid. The bone structure is totally different of a platypus. I mean, there's convergent in the sense they're kind of similar and they kind of serve a similar function in some cases, but you can always tell them apart. There's not like a full duck bill on a platypus.
But we have platypus today, right?
Yeah, that's right.
So we can do breeding tests. We have their genetics. We have the genetics of the platypus.
Right.
Okay. So what about in the fossil record, since all the examples, correct me if I'm wrong, all the examples that you presented in your opening statement were from the fossil record and therefore had no genetics associated. Like, for example, Tiktaalik or from the fish to tetrapod series, do we have any of their genetics at all?
No, we can't. That's a testament to how old they are.
And that's,
I disagree with that one.
And that's the problem that I have. For example, we have we could do 6 pages worth of examples of convergent evolution. Similar physical mechanisms are used to transmit sound waves from the ear to the brain in both vertebrates and certain insects. So we have these highly complex features having to evolve sometimes 30 times, like when it comes to the eye. I mean, how many times apparently did the eye have to evolve? So the question is, if we don't have the genetics, if we don't have breeding test capabilities in the fossil record, how are you going to be able to tell the difference? Just using a fossil example, the difference between convergent evolution, right? The evolution of these features independently, but yet they're still similar, okay, but not related, at least recent relationship, homologous. How do you tell the difference just looking at a fossil?
Well, it would just come down to anatomy, and maybe you're right. Sometimes maybe they would get something mixed up. But with I've cataloged like 2,500 transitional species or genera actually. The odds are that one of them at least is actually a transitional fossil, if evolution is true, which I know is the kind of a false premise in your view. But
well, here's a problem I have. Well, I'm sorry. Go ahead, Jackson.
I want to address the tetrapod thing. You mentioned Tiktaalik.
Yeah.
There were footprints found 10 million years earlier than that, but that doesn't necessarily mean Tiktaalik is not on the tree. It's a branch of the tree. That's all it means. And the foot, let me share my screen real quick.
Yeah.
The foot of the tetrapod that was found looks very similar to something like Ichthyostega. This was the footprint. These are the footprints. And this is Ichthyostega. So we have a very primitive foot with multiple extra fingers, so to speak. So it's not like we're finding a deer print, 10 million years before we find Tiktaalik.
So with these tetrapod foot tracks, okay, so here's a little more detail on it for the audience sake too. In 2010, it was reported that a well-preserved and securely dated landwalker tetrapod tracks from an ancient Polish marine tidal flat were found that were supposedly 397 million years old. Since these tracks would have been made before any of the creatures discussed in this article, and that would be the examples you look to, Ichthyostega, Acanthostega, including Tiktaalik. Right. The problem is, according to your model, right, Tiktaalik would be on the tree somewhere as a so-called transitional form,
but not necessarily the ancestor.
Right, not necessarily the ancestor. But remember, if we're comparing models and we're looking at agnostic lines of evidence versus lines of evidence that can differentiate between the models, this is not a problem for the creation model. Because again, we expect interesting mosaics, as I demonstrated in my opening statement. Okay, Tiktaalik being just that. We could look to Archaeopteryx, we can look to a platypus today, we can look to a seal, all sorts of different creatures. The problem is, for your specific model, okay, we've got tetrapod foot tracks before Tiktaalik.
Mm-hmm.
So one could say from the creationist perspective is we've got another interesting creature, just like we see all throughout the fossil record, just like we see today. It doesn't have to be interpreted as something that's transitioning into a tetrapod. If the tetrapod foot tracks are already there, pre-existing Tiktaalik, how's that a fulfilled prediction?
Well, here's the thing. Lobe-finned fish and stegocephalians are a very diverse group. We know about probably 100 species of both combined, and there are probably hundreds more we don't know about. They were distributed from the stegocephalians, like Tiktaalik. They were distributed from Africa, Europe, Russia, Greenland, Canada, and it's not likely that we found the exact line that led up to what we know today. And
when it comes to the lobe-finned go ahead, take your time.
I remembered. Lobe-finned fish evolved in the Silurian period. If you find a stegocephalian footprint before that, then there's a major problem. But there is, like you said, the fossil record's not complete, and that's what I would chalk that up to. But we haven't even found what made the track, the skeleton of what made the track.
So I would say the evolutionist has faith that hopefully in the future they find more examples in order that the fossil record can become more complete. But when it comes to these lobe-finned fishes that you're referring to, we see that they first appear suddenly in the beginning of the Devonian rocks, along with many other types of fish with no evolutionary precursors. But the point that I want to make here especially is that we have a coelacanth. Okay, the oldest coelacanth fossil is dated by evolutionists at about 408 million years ago, right? While the coelacanth was originally thought to have been extinct, we now know that it's alive and well and adapted to live deep in the ocean. So lo and behold, today we have an example of a lobe-finned fish that at one point I believe was used, or maybe even still being used, as an index fossil, but today is a coelacanth evolving into anything other than a lobe-finned fish, Jackson, in your opinion, the coelacanth we have today, which I know there's some differences between the coelacanth today and the coelacanth in the fossil record, which we would say is the pre-flood world. That's expected, of course. We believe in change. But yeah, is the coelacanth today evolving into anything novel? Anything that's going to walk on land? Go ahead, take your time.
No, it's not. And there are basically two clades of lobe-finned fish. Coelacanth is in the clade that's not as similar to tetrapods as the tetrapodomorph fish were. Like Goganasis had a very tetrapod-like skull. It had spiracles for breathing air. Coelacanth doesn't have anything like that. Its teeth aren't anything similar to tetrapods. Its skull isn't really similar. It doesn't have spiracles. So it's a lobe-finned fish, but it's not really. It wasn't really ever considered to be an ancestor. And their fossils have been found not only early, but they've also been found in like the Cretaceous and leading up. So there is a fossil history of them. They're not just in the Devonian and then now they're present and there's no trace back in time, so to speak.
I would say, to at least conclude this one topic, because I do want to move to a couple other things you said. I think that the ultimate death knell here, just like in the article, for this story from fish to tetrapod evolution is what we were discussing earlier, these terrestrial tetrapod tracks that were found in rocks dated older than when any of these so-called transitional forms supposedly existed, because we, from the Cretaceous model, we would predict overlap, we would predict, and we see this in the fossil record, we would predict that the pre-flood world was a lot more diverse. And as I indicated in my opening statement, our model would suggest that we should be able to get a sense or an idea into how God designed the biological world and biological systems based on the way we design. And my question to you is, if you're familiar with these interesting crossover vehicles that seem to resist classification, and please, Jackson, don't say that these are not biological organisms, they don't reproduce. Listen, adding reproduction is even more of a problem for large-scale evolution, okay? So our model would suggest we should be able to get a sense for how God designed the biological world based on the way we design things, and we design interesting mosaics as well. We design what you would say is the perfect transitional form, as in your lines of evidence are agnostic to the debate, because what we see in the fossil record, what we see today is also reflected in the designed world based on the way human engineers design. My question to you would be, with these crossover SUVs, I mean, is this right here, is this a Jeep or a truck? Is this an SUV or a van? Is this Dodge Journey an SUV or a van? Can you classify these for me, Jackson?
Well, you know, if there is a God and he is guiding this process, this is actually a good analogy, because cars didn't just suddenly become cars. They were wheeling around carts not 1,000 years ago, and they technically evolved by the creator to these higher forms. So you could say that God is guiding the process in that sense, but I think God could be more creative if he wanted to be. I think he could put something out of a nested hierarchy or something that doesn't fit in the phylogeny. I think, of course, he could do it the way you described, but why would he? I would be as creative as I would want to be if I was creating something like that.
Well, we do see a sort of descent with modification with cars. Let's say you go to the earliest Chevy Cruze and the newest Chevy Cruze in 2022, but each version, each model was designed, right? You could say there's an evolution in
I'm not
the
necessarily
design.
saying they weren't designed. per se. But
now here's the thing: we as humans, we're just catching up in terms of design and complexity. We're just catching up to God, who understood this since the beginning. I mean, that's why we find these interesting mosaics. That's why we find that creatures have this built-in capacity to adapt based on environmental conditions and stimuli and stressors. For example, epigenetics, this extra layer of incredibly complex information where you have millions and millions of genetic switches just waiting to be turned on. Or through mutation, sometimes these switches are broken or permanently shut off, which temporarily usually has a type of advantage, even though it's overall reductive. So, I mean, the point is, God, his design in the biological world is a billion times more complicated and complex than us. I mean, today in 2022, we're just starting to design these types of modes of transportation or computer systems or cell phones. I mean, look at your cell phone. It has so many built-in capabilities where you get all these different apps, and it's all front-loaded into your cell phone device, things that God already thought of, knew of, and designed the biological world with. Like, how do you, from an evolutionary perspective, explain through evolutionary mechanisms like selection, mutation, the forward-thinking mechanisms?
Well, here's a good analogy.
Yeah,
with the phone. Sure, the phone is there, and you could say it's front-loaded, but it's getting new updates all the time. You know, like new genetic information. I know you guys aren't big on the concept of new genetic information, but like de novo gene birth. But I've got some notes here if you want me to read them about that.
Sure.
There was a study on stickleback fish on ancient genes, novel genes, and newer genes. They can tell the difference between them because the younger genes are expressed less broadly and at lower levels. 38% of ancient genes were in all populations, while 17% of the new genes were. New genes were less CG depleted than cytosine guanine depleted than ancient genes. Younger genes were often gonad specific. Young genes are expressed in one sex and one gonad. New genes tend to overlap with copy number variations, etc. So, how would you explain that away? That
now, are you saying that these are de novo genes, the origin of like orphan genes?
Right.
Okay, so is this like an example, like the fish fly example that I showed? And here, I'll go to the orphan gene examples here. Is this like the, for example, they've done tests on fruit flies, and they've realized that in water fleas, so here's an example of the water fleas that contain many orphan genes that are only expressed under specific sets of environmental conditions and stresses related to water availability. So the point is, you can have examples of fish, you can have examples of water fleas, different types of creatures that have sets of orphan genes that are not expressed, but then bring in a stressor or put them in a different environment, and these orphan genes that are already there, they're pre-existing in the genome of the living organism, now they're expressed. They're being expressed. They're just like pseudogenes, for example. Many non-coding RNA genes like pseudogenes are only expressed under certain conditions, which means the researchers there analyzing this data, they're not actually seeing the de novo formation of a new gene, a highly essential and functional gene. Okay, no, they're just seeing the expression of a pre-existing gene. I mean, how is that explaining the evolution or the creation of the gene itself? Go ahead.
Well, it's just a gene made from a non-coding section of DNA, and they can. I don't know if they've ever seen a de novo gene being born, but they can tell young from old. And that's a pretty good indication that some of them formed more recently than others.
See, what they look to is either gene duplication. They'll look to, you get a duplicated region of the genome, and then the duplicated region undergoes what's called neofunctionalization. You get a series of hopefully beneficial mutations, and then you get a new function out of it. For one, there's a waiting time problem for that. We've never seen it. Yeah, as you're putting forward, they're sequencing these genomes, analyzing these genomes, looking at similarities. They're seeing a lot of orphan genes now being expressed, trying to say that, you know, this is an example of a new orphan gene popping up. They oftentimes see the loss of orphan genes. We know speciation events, okay, variations within the same type. We know that these oftentimes occur due to reductions in genetic variability, allelic variability, shifts in heterozygosity to homozygosity. So by definition, you are going to lose variation. You are going to lose orphan genes. That doesn't explain the de novo origin of these orphan genes. And notice here, orphan genes are being demonstrated to have key roles in a variety of complex genetic and biochemical pathways in a variety of creatures across the spectrum of life. So this idea of de novo gene synthesis, this is the last thing I'll say, and I'll get your response, is it's circular. Okay, the evolutionists, the naturalists, they see these taxonomically restricted and essential genes, which is exactly what we would predict from the creation model. They see these, so they think, well, how did these come about? They must have come about through evolutionary processes. So they must have come about through non-coding areas of our genome, right? De novo gene synthesis. This is circular because all they're saying is, well, you know, it couldn't have been created that way or designed that way. So why do orphan genes exist in the mind of the evolutionist? Is, well, because of de novo gene synthesis. But then why is there a process called de novo gene synthesis? Well, because of orphan genes. You see how circular that is? Instead of just admitting, no, these are fully integrated into the information systems of life. They're highly functional, highly essential. They couldn't have evolved. We haven't seen them evolve in the lab. Okay, observing the expression of a gene that's not been expressed, observing the loss of a gene, that's not explaining the origin of a gene. It's much more likely to lose a gene or that a gene will be broken down through a series of mutations than for a gene to be built up. I said a lot there, but yeah, what are your thoughts? How do you get around the circularity of de novo gene synthesis? And do we have any evidence of this gene duplication and neo-functionalization in terms of orphan genes? Go ahead, take your time.
Well, I'm not saying that epigenetics isn't a thing, or that orphan genes or pseudogenes don't have a function. Oftentimes they do. What was the rest you said? That was kind of a lot.
Well, how do you get around the circularity of de novo gene synthesis? We don't observe it today in a lab.
Right. They haven't observed it, I don't think, but they can infer it. They're not just saying, well, orphan genes exist, therefore they must have been created out of nothing. I think there's more to it than that. I mean, they're seeing signs that some genes are younger than others based on the cytosine, guanine, what's the word? I forget. But
right, they're just comparing
and they're more gonad specific, the younger ones.
Yeah, they're comparing similar sequences, right? For example, the whole point, especially for the audience, they call them orphan genes because they have no ancestral sequences in other creatures that they evolved from. So they're looking at similarities in what the evolutionists believe is shared relationship, right? And they're hoping that, well, there's some similarities here. So maybe this is the precursor to the orphan gene. Maybe this is the precursor in the non-coding regions of the genome. Point is, it's not empirical. They're not actually seeing, just like endogenous retroviruses, for example. Can you present me with a technical paper of a non-functional endogenous retrovirus or any type of retrotransposon that has gone from something that is non-functional to something incredibly and critically functional in the genome? For example, we know ERVs are functional in determining cell type, cell stress responses. ERVs are functional in the embryo. I mean, do you have any example of this being observed in the lab, this type of co-option?
I would have to do more reading on that. I don't really have an answer to that one. But again, what was the first part? I keep losing track.
Well, I'll just break it down like this. How do you get around the circularity? I understand you believe that de novo gene synthesis explains the existence of orphan genes. You believe that. But we haven't observed it. It's not empirical. It's inference. And I don't even think it's a good inference. How do you get around the circularity of that?
Oh, you're talking about ERVs. That's what I was going to talk about.
Right. Okay, go ahead.
5 minutes.
Sorry to change the subject, but ERVs, you say, I think your model is that viruses came from the DNA, and that's why they're endogenous retroviruses. They didn't graft themselves into it; they came out of it, right? Is that right?
Yeah. The question when it comes to ERVs and other classes of retrotransposons, including these ALU sequences, is are these created units of DNA function, or are they the result of past viral infections millions and millions of years ago? Now, I've shown numerous papers. For example, here's a bunch. Scientists identify new beneficial function of endogenous retroviruses in immune response. There's another one that has to do with regulation. The ALUs influence alternative splicing. Okay, regulatory activities of transposable elements. The pervasive retrotransposons that populate the genome have a powerful capacity to influence genes and chromatin, so on and so forth. Retroviral promoters in the human genome. So the point is, the prediction of the creation model is these were designed this way. There's obviously been rogue elements, mutations, things like that. Viruses in general are good, right? Healthy. They regulate the amount of bacterial species in our genome, for example. So that is the question, and we find function. And so I think the evidence best fits with the creation model. Yeah, go ahead. That's how we would explain like ERVs and retrotransposons. They're initially created, designed that way.
Yeah, but I think I've heard you say that viruses came from those segments of DNA. Is that right, or am I misrepresenting you?
Because, for example, I asked you this question, I think maybe last week. Since a virus requires a host to replicate, right? Mm-hmm. What came first, the virus or the viral-like element or the host?
Right, the host probably would have come first.
Okay. And that's exactly what we're saying. And there's a model put forth by creationist PhDs. I wonder if I can find it here, but I got a million things here. So where is it? Okay. So the creation model. It is possible to reinterpret retroviruses. Okay. Accordingly, retroviruses could have originated from within the host genome. Okay. You admitted that the host would have predated the virus. Okay, that's exactly what the variation-inducing genetic element hypothesis put forth by creationists suggests. Okay, accordingly, retroviruses could have originated from within the host genome instead of invading it from outside. This had to have been the case in any scenario since viruses are dependent upon host cells. This means that the cell's genome had to predate the viruses. You can read all this yourself. The point is, we find confirmation of this model and hypothesis based on the function that we find in paper after paper after paper when it comes to ALU sequences, endogenous retroviruses, all different types and classes of retrotransposons. So for your model and your explanation to have any validity, you'd have to show me how it's possible for endogenous retroviruses to go from non-functional to something extremely functional. Does that make any sense? And take your time, Jackson.
Yeah, well here's the thing about your model. You couldn't make a virus out of the ERV most of the time if you wanted to, because it's so fragmented. They're fragments of a viral genome. They're not the whole viral genome. So you couldn't really, that wouldn't really work most of the time anyway. Maybe there's some hole, but from what I've read, it's pretty fragmentary.
Are you saying that the only parts of the ERV are functional?
No, I'm saying only parts of the ERV viral genome is there, like compared to viruses. I'm not saying it's only partially functional. I'm saying it's there. Some of them do have functions. I'm saying you couldn't make a virus from that if you wanted to.
See, here's the thing. We have just viruses in general. We have more viruses in and on our body than we have cells, and we have 100 trillion cells. I mean, that's amazing to think about, right?
Yeah.
And what are the viruses there for? They're there for beneficial purposes. They help regulate the amount of bacteria and bacterial species that we have in and on our bodies. They're good. Okay, when people hear the word virus, they think of something bad. Only very few viruses are actually bad and harmful, and usually it's when a virus or a viral-like element leaves its original host or its original environment, right? So the viruses that were created or that exist in our body that are beneficial, if they were to jump to another species and that other species can't regulate it, it can become harmful to that species. And oftentimes viral-like elements can break off in our genome and become rogue elements. mutations. And that's what we're saying happened with these endogenous retroviruses and these transposable elements, like within the host genome, within these ERVs, you've got like the GAG and POL genes, which are functional to help with variation, things like this. But I don't want to spend all the time on this. I'll give you the last word on that. And then I definitely want to ask you a question that has to do with the rooted versus unrooted tree, because you made that argument in your rebuttal. So go ahead, take your time on the ERVs, have the last word on that specific topic, and then we'll move to this. I'm
kind of ERV'd out. Plus, I was reading the side chat, so I kind of lost track. Honestly, we can go to this point you're about to make.
Okay, so you made the claim that, well, this is an unrooted tree. Mm-hmm. It should be rooted, right? But here's the problem, Jackson, is whether it's rooted or unrooted, that doesn't change the data. Okay, we still know that the mutation rate in the mitochondrial DNA, as well as in the Y chromosome, is incredibly fast. We know that there's incredibly low variation in both mitochondrial DNA and Y chromosome. Every single Y chromosome on the planet, male Y chromosome, is nearly identical. And you can take every single Y chromosome, build a family tree, and that family tree reveals that we have all descended from one single Y chromosomal ancestor in the not so distant past, okay? And the same thing goes with the mitochondrial DNA. The reason why Dr. Robert Carter, okay, represents worldwide mitochondrial DNA diversity in an unrooted tree is because it gives a natural reflection or a natural reading or representation of the data. It allows the branches to flow out naturally, and that's why we see explosive growth from a center point. So why do you believe this whole unrooted versus rooted tree is an issue? Go ahead, take your time.
Well, the first part of what you said, I'm trying to remember, but I should have been taking notes. Repeat yourself a little bit.
It was the unrooted tree part.
Yeah, I know that. But
well, the first thing I said, well, right here. Here's an example I like to, show the audience, show you all my sources. The human Y chromosome exhibits surprisingly low levels of genetic diversity.
Yeah, that's what I was going to say. Everything I've ever read about mitochondrial mutation rates say it's incredibly slow. It's like one for every 3,600 years. It was just one kind of creationist experiment that said it was faster.
Well, the latest mutation rate studies, for example, one in the D loop region, the control region of the mitochondrial DNA, they discovered it's about 0.5 per generation. Even if you went with 0.2 per generation, that's still too fast. You can go to the Parsons study, which demonstrated that if you were to take that mutation rate back, mitochondrial leave would go just 6,000 years. A high observed substitution rate in the human mitochondrial DNA control region. It's pretty well established that the mutation rate, we can do pedigree stuff. We can take great grandmother, grandmother, mother and daughter, and we can sequence their genomes and we can find DNA differences and we can determine a mutation rate, the pedigree-based mutation rate. What you're talking about is the phylogenetic rate, which calibrates the known or empirical rate from pedigree-based studies. The empirical method, it calibrates the data, okay, to the geological column, to evolutionary-based assumptions. But I pointed out, I think I had time in my opening statement, is the evolutionists can't make those assumptions, because if you notice on this tree, go to the most common haplogroup in Europe, the HVR haplogroup. Notice the differing sizes of the branches. Well, what that means is people in the same time that go back to a common ancestor, they're picking up more mutations than their cousins. That means you can't make assumptions based on a straightforward molecular clock, because this is chaotic. This is just DNA differences, mutations over a lot of time. And you can't then make assumptions that the evolutionists do about when humans and chimpanzees split from a common ancestor, let's say between 7 and 13 million years. Okay, because what they'll do is they'll look at the amount of DNA differences that separate humans and chimps, and then they'll derive a mutation rate, and that mutation rate is very slow. But that's ignoring the observed mutation rate from pedigree-based studies is very fast. I said a lot there, but go ahead, Jackson.
Well, you mentioned they're correlating it with the geologic column. I don't understand how they're doing that.
Well, because if you, for example, we put out a video recently, Biblical Creation Basics, on mitochondrial DNA, and I think it was PBS. They put out a video where they admitted that they calibrate the data with what they believe is evolutionary history through the geological column. They'll oftentimes calibrate it with archaeology, right? They're not looking at the empirical method. They're not looking at known mutation rates from pedigree-based studies. No, they are calibrating it with their assumed history in the geological column. But here's the thing, it's the geological column that is in question. So that's circular. You can't refute the young earth creation model by calibrating the data with your geological column assumptions or your assumptions behind the human and chimpanzee split. That's circular. That's what we're debating here, is the existence of deep time. So we as creationists, we're just looking at the known mutation rate and we're like, wow, the known mutation rate through pedigree-based studies is fast. And we know there's extremely low genetic variation in the mitochondrial DNA. For example, here's a question I'll ask, and then please just take as much time as you need. I've been talking a lot here, Jackson. I don't want to dominate. So just take as much time as you want to address it. This is only a reflection of a few hundred generations, because on average, you got 20 to 25 DNA differences separating any two people in the mitochondrial DNA. Max, you've got maybe 120 if we were to include your longest lines, like the Africans. So with a fast known mutation rate, without any calibration, without any evolutionary assumptions, how do you get hundreds of thousands of years out of this? Go ahead.
I don't know, but I'm not sure that they calibrate it the way you're saying. I know that like some things do line up, like the Australopithecus seems to line up about the time when the chimpanzee human split happened. But yeah, I don't have much of a response to that. But
okay, that's
other than to say that when it's rooted, it looks a lot different.
Well, see, here's the thing. They root it, though, based on evolutionary assumptions. Where they're picking the root is based on evolutionary assumptions. So it doesn't give a natural reading of the data. Whether we root it or unroot it doesn't change the data itself. It doesn't change the mutation rate. It doesn't change the diversity, the low variation. It doesn't change anything like that. Unrooting it just gives us a natural reading or representation of the data. But we'll leave that up to the audience. With our last few minutes here, Jackson, I do want to ask you, because this is another thing you brought up in your rebuttal. And I think we do need a few extra minutes in discussion time because there's so many points brought up and we're discussing most of them. This is good. Your explanation for the chimpanzee and human Y chromosomes, the reason why they're so different, they're about 70% the same, okay, to the audience. But when you actually consider overall size differences, the fact that the Y chromosomes between humans and chimpanzees are only about half the size, when you consider overall gene content and architecture, it's actually only about 35% the same. So 70% the same, but when you consider everything, it's only about 35% the same. Okay, not to mention that the human male Y chromosomes have incredibly low variation and the mutation rate is fast. So how do you explain this data? And can you reiterate your argument in the rebuttal?
Yeah, the Y chromosome is a kind of the front line of reproduction and fitness, obviously. So it's going to be subject to mutations. And apparently, to answer another thing about the size difference, apparently chimpanzees suffered a lot of gene death in the Y chromosome and a lot of substitution. And yeah, I mean, again, the pan genus relies on a lot of males inseminating one female, and the fittest sperm inseminates the egg, or fertilizes the egg, rather. So that's kind of a reiteration of the argument.
Okay, so
kind of an answer.
So here's the thing. I've debated numerous people, including PhDs, Dr. Stephan Frello, I've debated, Gutzit Gibbon numerous times, Dr. Ron Garrett. They never have an answer to this. I think this line of evidence, the fact that humans, the chimpanzee and human Y chromosomes are remarkably divergent in structure and gene content, I think this alone falsifies universal common ancestry, because there's no answer. There's just so stories a lot of imagination in terms of the answer to this. And I've seen this with people like Guts Gibbon. They just dodge, duck, dip, dive, and dodge. I'm not saying that you're dodging, but here's the problem is the Y chromosome and the mitochondrial DNA, they are our uniparentally inherited DNA compartments. They're non-recombining, okay? In the Y chromosome, for the most part, they're non-recombining. That means they should be the most stable. That's actually why we look to the uniparentally DNA compartments, inherited compartments, to build these family trees that go back to one Y chromosomal ancestor, one mitochondrial DNA ancestor, who, by the way, are nothing like the chimpanzee. Nothing like the chimpanzee. They should have been slightly similar to the chimpanzee. But here's the thing. Because there's no recombination, they should be more stable. And yet, the Y chromosome was the most different between humans and chimpanzees. As a matter of fact, and address this one, the human Y chromosome and the gorilla Y chromosome is more similar than the human Y chromosome is to the chimpanzee Y chromosome. But wait a minute. The evolutionary story is that humans and chimpanzees split from a common ancestor 6 to 12 million years ago. And we have a more distant common ancestor with the gorilla. Why is the human Y chromosome more similar to the gorilla Y chromosome? And this is the last thing I'll say, and then you can yield, because this argument that the chimpanzee is kind of the odd man out and there's a lot of sperm competition, and therefore there's been more change and loss in terms of degeneration than the chimpanzee Y chromosome. They're the odd man out kind of thing. This is all nice. This is all cute. But without any numbers, without any data, without anything empirical, it's just a story. And Dr. Rob Carter points this out. And then I want you to take as much time as you need to respond. What are the implications of this new information? First, for the evolutionists, the Y chromosomes must be evolving much more quickly than anyone imagined previously. They are now going to have to apply. Notice this, Jackson. So when you respond, I want you to give me this data. I want an empirical answer. They are now going to have to apply, and this is something none of my critics have ever done, including the Gutsy Gibbons of the world, have to apply mathematical models to try to demonstrate how a sequence can change extremely rapidly. You can go with your chimpanzee sperm competition. Go with it. Show me how mathematically, empirically, we can get wholesale rearrangements of significant parts and the evolution of entirely new gene families in a relatively short amount of time, yet stay homogeneous. This is important, yet stay homogeneous within a species. Go ahead, take your time, Jackson.
All right, that was a lot. But first I'll say the gorilla and chimp and human Y chromosomes, I think the reason that the gorilla would appear more close to us is because the chimp has gone through so much change. And with gorillas, they have a male-dominated society and multiple females, so there's not as much sperm competition. So that might give the appearance the Y chromosome of a gorilla looks like it's closer to us than the chimp is, because the gorilla is our next closest relative.
But are you done?
I forget the last part. I'm on a lot of gabapentin, so it's hard to remember stuff.
No worries. You're doing great. This is an awesome discussion. But here's the thing, Jackson. The evolutionists are always asking for data that contradicts phylogenetics, nested hierarchical patterns. Here we have a Y chromosome, which should be more stable. That's way more different, okay, in the humans and chimpanzees than between humans and gorillas. And again, the last thing I asked you for your response is, I don't want a story. Like, I know you believe that in terms of the chimpanzee, they're the odd man out, right? But as Dr. Robert Carter pointed out, you have to demonstrate this through empirical data. You have to apply this explanation, which hasn't been done, because the last paper I read on it, they admitted this is just a hypothesis. So they believe that this explains the incredible divergence between chimpanzee and human Y chromosome, but without the application of a mathematical model to demonstrate how these sequences in the Y chromosome can become so different, it's just a story. So go ahead. What are your thoughts on that?
Honestly, I have no answer, but I will look into it. And, I've been doing a lot of math with this virus stuff. I'll look into that, honestly. So, I'm not a geneticist. I just got into genetics recently, doing some research off and on. Been doing a lot of viral research for this genetic entropy stuff. So I don't have a good answer, honestly.
No, that's okay.
The honest answer, yeah.
But I do want to say this, Jackson, before Matt jumps in. The debate is all life related through common ancestry? I think the genetic data, which is the best way to determine ancestry. Okay, we don't even have to go as far as whales and pine trees. The evolutionists like R and Rob, they always say, oh, these creations, they just want to go like, the two furthest forms of life on the, tree of life and kind of make fun of it. No, let's go as close to home as possible then. Humans and chimpanzees. And guess what? The evolutionists didn't predict this amount of difference. They didn't predict that the human Y and the gorilla Y would be more similar than the human Y and the chimpanzee Y. It makes sense from our position because all genetic data suggests separate ancestry. God created Adam and Eve independently from every other form of life. We have a confirmed expectation and prediction here. We've got the Y chromosome that fits perfectly with our model. We don't have to make up any fanciful stories. It's the evolutionist that has the problem. And I can assure you, Jackson, when you look into it, there's been no sophisticated mathematical modeling being done. So if you find out that there's been no sophisticated mathematical modeling being done in terms of this Y chromosome problem, what's your conclusion going to be?
It's going to be that, well, first of all, let me ask you this. Why would we be similar to gorillas at all? I mean, well,
here's the thing. We are all made of DNA, RNA, protein, right?
Sure, but
DNA,
why are we
RNA,
similar to the things that appear to have, through the fossil record and other means?
Well, what's more similar? What's more similar? A hatchback and a sedan, or a hatchback and a boat?
Right, but they don't have a DNA code that says they're very similar.
Well, they have a blueprint. They have underlying materials that are used, to design and engineer and build the products. But here's the thing: adding DNA, adding reproduction, that's just adding more complexity. Imagine we could do that.
Sure, it's very
Imagine complex. We could do that. But my point is, just answer what is more? Why is a sedan more similar to an SUV than a sedan is to a skateboard?
I don't know how to answer that, honestly. I mean, I'm kind of confused by it.
Well, because there's just more similarities in the way that the sedan is designed and the SUV is designed than between the sedan and the skateboard. By definition, when God created life, he had to create, Jackson, some creatures that are more similar to other creatures. For example, humans and chimpanzees, humans and gorillas, humans and bonobos, humans and...
But why do you have to do that?
Well, if we go back to our model at the beginning, do you remember what I said? The Bible says that we are made in the image of God. Therefore, there should be something that reflects us, that reflects the divine. But us, that reflects the divine if we're made in his image. So we've formulated a hypothesis or a model that suggests, okay, if we really are made in the image of God, maybe we can get a sense for how God designed the biological world based on the way we design things. And it just so happened to turn out, Jackson, that we build in homologous patterns. We build in nested hierarchical patterns. We build interesting transitional forms. And that's exactly what we see in the biological world. How do you explain that? It matched up so perfectly.
Well, I would just say that God created us in the same category as the filthy apes. I wouldn't have done that as God.
Well, wait a minute. But here's the thing. If chimpanzees didn't exist, Jackson, then the evolutionists would just say, well, humans and gorillas, look how similar they are.
Well, that would be the next closest common ancestry.
Yeah. And then remove gorillas. Oh, well, the evolutionists look at, humans and bonobos. Look how similar. And then remove bonobos. Well, look at humans and lemurs. Remove lemurs. Look at humans and, the old world monkeys or something. So it's never going to be good enough for the evolutionists. Are you trying to say that humans, answer this question, are you trying to say that humans should be equally distant in morphology, anatomy, genetics, and physiology, equally distant, okay, from all forms of life? You have humans and then everything else is equally distant. I mean, dogs, a banana plant, a chimpanzee, it's just equally distant to all three of those. Go ahead.
Well, not necessarily just humans. I think a lot of things should be distant from a lot of other things, and they're just not when you look right at it.
I would say it doesn't. I would say it's not very sophisticated thinking to say that if God designed the biological world, that we wouldn't expect more similarity between humans and chimpanzees, even before we had genetic sequencing technologies. Just stand back and look at the outward appearance of the organism. How many similarities do we have with the chimpanzee? You can easily predict, sure, you know, in the DNA, in the genotype, we're going to expect more similarity between humans and chimpanzees than between humans and a dog. Just stand back and look at the outward appearance. But here's the thing. Did evolutionists answer this question? Did evolutionists make a prediction as to how similar genetically humans and chimpanzees would be before we sequence their genomes?
Well, it used a little bit of circular reasoning there, too, to say, well, chimpanzees look similar to us. That means they must have DNA. And if we say, well, the DNA looks similar to us, that means they must look similar to us. That's kind of some circular reasoning there as well. But
well, how's that?
What?
I'm sorry. Go ahead. Answer the second question. Yeah, go ahead.
Again, I forget the second question. Can you repeat it?
Did the evolutionary community make a prediction on just how much similarity there would be in the genetics between humans and chimpanzees before we discovered the similarity?
I'm not sure if they did or not.
I don't think they did. Now, I could be corrected on that, but I'm pretty sure they didn't make a prediction, which means it's not evidence for evolution. They never made a prediction like, well, you know, humans and chimpanzees, they're going to be 99% similar, 97% similar, 95%. They never made a prediction. We sequenced their genomes. It turned out we're pretty similar, at least more similar with the chimpanzee than we are to
anything else. Yeah.
Now, if we didn't have these similarities, if we didn't have these nested hierarchical patterns, how could we ever advance in, let's say, the medical industry? I mean, when we're doing experiments, when we're looking for cures, when we're studying the biological world and finding out things about this amazing planet that we live on, if we didn't have these similarities in DNA, RNA, protein, so on and so forth, how would we ever advance? I mean, what do we use mice for, Jackson? I mean, don't we use...
I use them to feed snakes.
Right. But like, you know, without the similarities in even mice, you know, and the evolutionists will say, well, the reason why humans and chimpanzees share more than humans and mice is because, you know, humans and mice have a much more distant common ancestor. That's where they look to the DNA differences. But I would say the lack of uniqueness displayed in things like incomplete lineage sorting, where you can look at different genes and come up with totally different trees, convergent evolution, loss of traits evolution, you know, these types of things that demonstrate a lack of uniqueness in the phylogenetic tree. It's nothing but a rescue device and a story from the evolutionists when it comes to the inconsistencies. They rarely want to focus on the inconsistencies. Jackson, have the last word. It looks like we've already gone 15 minutes over the discussion time. Have the last word in the discussion, my good man.
Well,
I said a lot there.
I'll just say that was a lot, but I'll just say you're basically saying everything was created with DNA hierarchies and anatomical hierarchies for future medical research, looking forward thousands of years. And I don't find that a very good explanation, but I mean, we all have our own conclusions, so.
You can have the last word, Jackson.
That's it.
All right, then. We are going to jump into our closing statements, which are about 5 minutes long, and then go right into Q&A. So, who is going first on the closing statement?
I guess it's me.
Like
you zoomed in on me.
Yep. Looks like you're up on this one. Okay, 5 minutes
All
whenever you're ready.
Good debate, good discussion. I love doing this stuff. I forget 90% of what I say during the debate, so I don't know how I did at all. I might have done terribly, might have done great. I have no idea because, again, I'm on a lot of Gavin. That's the joke I like to use. But yeah, Donnie and I are friends, and nothing will change that, I don't think. But I do say the evidence points to common ancestry, and you know, there are a lot of stories about, well, cars are built in hierarchy, so they must have been created. Well, why would God, in His infinite wisdom and infinite creativity, need to do that or want to do that? Surely He would choose at least 2 separate lineages and kind of build from there. Why would He just do all one? So, yeah, good debate. I'll be in the after show. And again, contact your senators about that exotic animal ban. Let's not let that pass, but not like contacting your senator ever does anything. So I'll hand it back to Donnie.
All right, Jackson. And if you have a link to what you just said there at the very end, you could post that for the audience below so we can let them know.
Yeah.
Take a sign. All right, and Donnie, when you are ready.
All right, let me get my timer here. 5 minutes. And I appreciate that, Jackson. We are friends. We are good friends, and I appreciate this profitable discussion. And I always appreciate how cordial and respectful you are. And I appreciate you asking the good questions. So, okay, I am going to start my timer here and share screen. So, again, universal ancestry or separate ancestry. Jackson's burden of proof, the burden of proof for all the evolutionists. Don't show us variation within a kind. Okay, and you can define a kind as all those organisms that go back to an ancestor. And you know, we've done that with Dinosauria. We've shown created groups. We've also done that with, especially with humans. The Australopithecines, there's a separate ancestry model, but it's their burden of proof to demonstrate that not only dogs and wolves are related, but that dogs, wolves, and banana plants are related. Okay, homology is not going to do it. Nested hierarchical patterns is not going to do it. Transitional forms is not going to do it. And the evidence, the best differentiating lines of evidence that we discussed today, and this is my 95th debate, and I've had debates on all sorts of topics, but these main questions that I have regarding the Y chromosome data, we didn't even get into the hat map data. I didn't have time to get into the linkage block data that suggests separate ancestry, because we have these massive linkage blocks that have no evidence of recombination gene conversion. I mean, there's some, but not nearly enough. If we have recombination every generation, let's say, okay, you're scrambling up the DNA variants for hundreds of thousands to millions of years, then why is there still so many large linkage blocks in the human genome? There should have been much more scrambling if human evolution were true. If we actually go back a couple 100,000 years. I've asked that question to PhDs. I've asked that question to your most militant evolutionists, and they never have an answer. I didn't see any answer to the satellite DNA problem from the Ventura paper when it comes to the chromosome 2 fusion. And when it comes to the chromosome 2 fusion, if there was no fusion, then guess what? There was no human evolution. And he talked about the centromere and the telomeres, and no, that's wrong. That's wrong. However, both the alleged fusion and centromere sites fail the test when analyzed closely. Not only are these alleged evolutionary signatures very small in size and muddled, degenerate, but both of them are found inside what? Functional genes. The alleged fusions site functions as a genetic switch called a promoter inside a long non-coding RNA gene, and the so-called fossil centromere, which can't be found, Jackson, contains both coding and non-coding sequence inside a large protein coding gene. Combined together, these data destroy the evolutionary hypothesis. And again, the question from the Ventura paper, in the fact that chimpanzee and gorilla chromosomes differ from human chromosomes by the presence of large blocks of subterminal heterochromatin thought to be composed primarily of arrays of tandem satellite sequences. Listen, the evidence refutes a chromosome 2 fusion. If there was one, then based on the satellite DNA observations that we have here. At best, you have a human-specific fusion, which can best be explained by the creation model, because we have two bottlenecks. We have the bottleneck at the flood. We also have a mini bottleneck at the Tower of Babel. So we could just go as close to home. Pseudogenes, functional DNA elements necessary to sustain healthy life processes in the cell, hierarchies. And last thing I'll say, why did God create this way? Well, he created this way for functional purposes. And this is exactly what we see. We predict that, so you got the universal ancestry model that suggests that the majority of the genome is junk, consists of genomic fossils, consists of evolutionary leftovers. And yes, if that's exactly what we found, that would be reflective of universal ancestry, but that's not what we find. Over 80% of the human genome is actively involved in at least one or more biochemical reactions associated with gene regulation in at least one cell type. Function after function, gene expression, regulation, function for pseudogenes, endogenous retroviruses, functional redundancy, these third position codon. The third position codon variation we now know is involved in information flow. It's not just there to tolerate error. They're there to help slow and speed the process. ALU elements, co-option doesn't work. What we're seeing is confirmation of the separate ancestry model. We predict function. The evolutionary model predicts junk, evolutionary leftovers and genomic fossils, and we don't find that. So why is there homology? Why is there nested hierarchical patterns? Why do we find interesting mosaics? Because they were designed that way for a purpose, for a functional reason, and that's exactly what we find. And we have people like Dr. Nathaniel Jensen who've made testable predictions based on this model of created heterozygosity that suggests most of the genome is functional. Most of the DNA elements in the genome, like pseudogenes, ALU sequences, and ERVs, are functional, and that's exactly what we find, contradictory to the universal common ancestry model. And the proponents of universal common ancestry are not stepping up to the plate and making better testable predictions. So
Time.
why, and how much time do I got?
Time.
There you go. And thank you, Jackson. I appreciate the debate. Tons of fun. 5 minutes flies by. So,
okay, everyone, we are now heading into the questions. So, Jackson, these are like 90% for you. So I'm sorry.
Yeah. Sorry.
But we are going to jump into the super chats first. So the first one is going to be from Logical, Probable, Possible. Thank you for the first super chat, directing everybody into your after show, which I will remind everybody again later. And then to Slew, I hope I got that name right. And it is for Jackson. It says, what explains the giraffe's neck better? Darwinian or Darwinianism or Lamarckism? And why would, I guess we could just start with that. It's a two-part question.
Yeah. I'm not good at remembering questions, as you can remember. But like I already forgot what that one was. No, Lamarckism doesn't work. It's not a thing. So it would be like neo-Darwinism, just a gradual process. It's not, the giraffe stretches its neck, so the next generation has a longer neck. That's basically Lamarckism. So that's not how it works.
And the second part, why would an organism change to begin with?
To adapt to a new environment.
All right.
Yeah.
Yeah, it was just a short question.
Yeah.
Next one was from Logical, Probable, Possible. I guess the question would be for you, Standing. Will Joel Duff join the after show?
There's a good question. Yeah, let's see if Joel Duff has any answers to these problems that have not yet been answered. Let's get an answer. And maybe some better testable predictions, since Joel Duff likes to attack models from people like, Dr. Nathaniel Jensen and Dr. Sanford. But he's a nice guy. So yeah, let's have him join the after show.
All righty. Next question. Question for Roe. Anthony from Anthony 1701. Man, he's really old. What is the best beneficial mutation you know of without any deleterious effects? And how will this mutation work against other mutations without being bred out after a few generations?
Like the gene that in the Linsky experiment that allowed E. coli to metabolize citrate was a duplication mutation. And how it would balance out with the other ones, I don't know. But yeah, they do add information, and so.
Okay, good.
Matt, let me respond.
Yes.
So when it comes to beneficial mutations, okay, they do occur, but there's a few problems with that. And the problems are that they occur infrequently. They're rare. I think it's about 1 in a million they've discovered from the Lenski experiment. And when they do occur, they're usually reductive and functionally compromising to the organism. Even a lot of their best beneficial mutations, it just turns out that it's a gene that's broken and permanently shut off. And there's a waiting time problem when it comes to these beneficial mutations becoming actually fixated in a population, because there's a lot of genetic noise. Oftentimes selection doesn't even see them. And when it comes to a lot of these beneficial mutations, like I believe the major one seen in the Lenski experiment, all we're seeing is rearrangement of pre-existing regulatory system. Okay. But that's not going to drive a fish to fisherman or bacteria-like biologist, a bacteria-like organism into a biologist. I mean, if you look at sickle cell anemia, it's got, I guess, a bit of an advantage, even though malaria is bad, sickle cell anemia is bad, and it's all due to a broken gene, broken protein, broken cell. How's that ever going to counterbalance the damage done by this low impact, nearly neutral, deleterious mutation accumulation? So the point is, beneficial mutations, although they happen, they're never going to drive large scale evolution. I yield.
Okay, I guess that will bring us into the next question again.
Well, I think if Jackson wants the last word, because it's his.
Not sorry. We can go to the next question.
It's on the same topic. So, if whales went from fully terrestrial Pachycetus, or I'm not sure what he actually said.
Pachycetus.
Yeah, Pachycetus, to fully aquatic Basilosaurus. In 200,000 generations. How can't E. coli evolve into literally anything at all? Well over 65,000 generations.
Well, I don't know how many generations it took for whales, but bacteria are reproducing at the rate they've always reproduced relative to them. I'm not sure if that makes sense. It makes sense to me in my head, but I'm not sure that translates out. So you're not going to just pop out a frog from some bacteria. That's basically my answer.
I'll respond, Matt. We'll just share a screen. It's funny because Lenski's bacterial populations, we've got thousands and thousands and thousands of generations of bacteria, and all we ever get is bacteria producing more bacteria, but the more bacteria is degenerated bacteria. So we got bacteria producing bacteria in these experiments, fruit flies producing fruit flies in the many thousands of fruit fly experiments. But those that believe in universal common ancestry want to say that fruit flies, bacteria, and strawberries are related. And that's why they can't meet that burden of proof, because that's not what we see empirically. When it comes to Lenski's experiment, I find it funny because Lenski, as you can see here from Dr. John Sanford, although a very trivial adaptation happened, optimal growth on a given medium, his bacteria shrank in genome size. The functional genome decreased. Evidently, the more rapid growth was largely accomplished through genetic degeneration. Many useful genes not essential in that artificial environment were apparently lost. When transferred to a natural environment, those highly degenerated bacteria would essentially be dead on arrival. So we have bacteria that are losing genes short-term for adaptive purposes, but what it actually is long-term degeneration. And the last thing I want to say in terms of the whale evolution is, even according to the evolutionist time frame, okay, millions of years is not enough time to convert a land mammal to a fully aquatic mammal. I mean, how does Jackson explain empirically these types of novel adaptations, emergence of blowhole and musculature and nerve control, emergence of ball vertebrate, modification of the eye for underwater vision, change in birthing process where fetus is delivered in breech position, ability to nurse young underwater, origin of blubber for temperature insulation. So, I mean, the origin of tail flukes and musculature. Again, this goes back to Jackson's opening statement and the opening statement of most well-studied evolutionists is, don't tell me that it happened if you can't tell me how it happened. If you can't tell me how it happened, then you're just imposing your presupposition on the genetic data, which is the empirical, direct way of determining ancestry. I yield.
Well, a lot of the physiological features were already present by Pagasitis, but not all of them, obviously. But like the birthing thing, myositis was found with the fetus inside it. It indicated it was still giving birth on land, even though it was semi-aquatic. And tail flukes, Paragasitis had a compressed tail caudal vertebra that would have given it like a kind of beaver-like tail, kind of an intermediate tail fluke. So that's all.
All right. Next question is going to be for you, Donny. It is, I think that the strength of Jackson's model is the ability to predict where fossils will be found. How does Standing for Truth explain their ability to predict where a specific fossil animal will be found?
Sure. Good question. Jackson and I discussed that at great length in the discussion portion. And again, Jackson, I want to thank you for a sophisticated discussion. We talked about so many different topics and subjects, and I think this was one of my favorite debates. So
sorry, my memory isn't that great, or it would have been a better debate.
That's okay. And you know what? Even the greatest memories can't always remember every single point that's being brought up in a discussion such as this, a highly technical discussion. So here's the thing that I find funny is they can't accurately predict the series of so-called unnecessary trends. Here's the thing. We would need thousands and thousands and thousands of so-called transitional forms. Okay. If evolution really were true, that we've got a single-celled-like ancestor billions of years ago, evolving into a multi-celled ancestor, into a fish, amphibian, reptile, mammal, okay, eventually a man, something as sophisticated and complex as a man, one single cell, more complicated than the space shuttle. Not to mention that single-celled ancestor billions of years ago would have had to come from non-living chemicals, which we have no evidence for, which is why the design diversity hypothesis is most reasonable and most plausible. Now, this predictions, no, they're failed predictions. Even the Tiktaalik example, and I understand that they can force fit it into the story. Well, it's still on the tree. It's not the direct ancestor. Okay, sure. You got a population of this creature called Tiktaalik. You don't know if that's just an evolutionary dead end, an interesting mosaic. You don't know that evolved into anything. You don't know that species survived far enough into the future to actually evolve into a land-dwelling tetrapod. No, what we find here is tetrapod foot tracks before Tiktaalik. That means we have tetrapods living before Tiktaalik, which is best understood from our model because that's what we believe is in the pre-flood world. We have overlap. We have different ecosystems, right? We have logical zonation. This is our model. When we apply the creationist global flood model of progressive burial by ecological zonation, the presence of these unusual fish-like fossils that we talked about that Jackson presented makes perfect sense since these creatures were buried in the flood layers shortly before most coastal land plants appear in the Carboniferous. It becomes apparent that they likely inhabited coastal waters based on their various unique adaptations and their place of burial in the flood strata. When it comes to the evidence, the fossils say no to tetrapod evolution. No, the predictions fail. They're not good. Anyways, go ahead. Jackson, you can respond if you want.
Well, this doesn't just apply to transitional fossils. It applies to pretty much any fossil. I mean, where are you going to find red-liched trilobites? In the Cambrian, without exception. Where are you going to find other species of trilobites? Up until the Permian and never past it, without exception. Where are you going to find Gorgonopsian therapsids? In the Permian. Where are you going to find cynodonts most of the time? The Triassic. Where are you going to find archosaurs most of the time? The Triassic. Where are you going to find there are 100 more examples I could go on about, but that's basically the answer.
Sure. I can have a final response. So again, that's assuming, and the places where you can actually find that sequence, okay, Precambrian, Cambrian, up to the Jurassic, Triassic, so on and so forth, Permian, okay, is less than 1%, less than 1% of the Earth strata. The entire Earth is where you find the so-called layers in that sequence, which means and you still don't get all the necessary strata that would have to be there if the geological column as portrayed in the textbooks is true. So that's why here we go. Does the geological column exist? I've got hundreds of slides here. So you can read through this where it shows, secondly, those locations where it has been possible to assign all 10 periods represent less than 0.4% of the Earth's surface, or 1% if the ocean basins are included. It does not engender confidence in the reality of the geological column when it is absent 99% of the time. So the point is, most of those layers were formed at the exact same time. We have amazing evidence for the flood. For example, the cold slabs that were predicted from catastrophic plate tectonics that are found deep within Earth's crust that should have melted if tectonic forces put them down there millions of years ago. The fact that they're still cool and they haven't yet either melted or warmed up to the surrounding material is fascinating evidence of the global flood and rapid subduction, and therefore rapid burial during the flood, ecological zonation. Remember, this is a burial during the flood. As the floodwaters inundated the continents, the shallow marine invertebrates were first swept from the pre-flood ocean floors and buried on the continents in rapid succession. After the waters rose over the continents, they progressively encountered different ecological zones at different elevations, which were inundated in rapid succession. So sure, you got different ecosystems, let's say with the trilobites, which we wouldn't expect to be living, let's say, with humans. We see different elevations in Genesis. We know that there are rivers flowing out of Eden, and we know rivers flow downhill. So we have different elevations, different ecosystems, humans and dinosaurs living in different systems. Okay, so the evolutionists, of course, are always going back to that assumption that the geological column exists. I think the question was for Jackson, so he can respond however he wants, but I want him to address, especially.
No, that was for you.
Oh, it was?
That was yours.
So, yep. Okay. I'll yield there.
All right. Question is then for Jackson. What would be an alternative explanation for the diversity of life if both creation and evolution turn out to be wrong?
That's a good question. I guess it would be a different God. That would be my best guess, but I don't know.
Donnie, what would you say?
So if creation and evolution wasn't true,
right, is there a third option?
I guess either we're created or not, created or evolved, or I guess there's a percentage of people out there that just say we're not really here. Maybe this is the Matrix, maybe this is some kind of computer simulation. So who knows?
Right. All right. Question for Donnie this time from DMC. What did animals eat after the flood if the earth was covered in salt water?
If the earth was covered in salt water after the flood, what did they eat? Well, it depends if the pre-flood world was fresh water and then gradually became saltier from the flood itself, the fountains of the great deep breaking open, leading and resulting into the perfect chemical composition of the water to form things like limestone, so on and so forth. So as the oceans gradually became saltier, we also know that the pre-flood world had incredible diversity in terms of plants and organisms and just fauna. So we would have plenty of plants that were uprooted, right? And then replanted either during the flood or after the flood, where you'd have tons of plants to eat, tons of fish. I don't see it being a problem if the salt water is just going to gradually accumulate during the flood and then after the flood. So there'd be plenty of plant material to eat. which would not be a problem for a lot of the pre-flood animals that were brought on board the ark. Those archetypes, many of them would have been vegetarian. Plus, Noah would have brought a ton of different types of food onto the ark for the animals themselves. So I don't see there being a problem.
Well, I see it as a problem because basically the whole world is leveled and all the vegetation would be destroyed. I don't see how a tree is going to get picked up and replanted somewhere else, which is kind of an oversimplification of what you said. But yeah, they get off the boat. There's nothing. It's a barren landscape. Maybe there's some dead plant material, badly decayed after a year. Just mats of goo to eat, maybe. But the herbivores don't have that problem because they're going to get eaten by the carnivores anyway. So, I mean, we kind of have a major problem.
Well, I guess I get the last word. So again, Noah could have brought on, which makes sense. I mean, he would have been incredibly smart. He had 100 years to build the ark. He would have brought on fodder animals, like, tortoises are a good fodder animal, for example. He could have brought on dried meats. Okay, tons of food to eat during the flood year, and also food that can be eaten afterwards. And we see it in floods today. Of course, if you have plants being ripped up, trees being ripped up, they can be rooted again after the fact. You'd have plenty of dead animals, plenty of food to eat. When it comes to the carnivores, apparently eating the herbivores, I think that has to do with a lot of the migration patterns where you have more of the predatory animals driving a lot of the non-predatory animals like kangaroos further and further. And that's where you see them getting stuck in places like Australia. You'd have land bridges where you'd have a post-flood ice age where you'd have the water levels are not raised as high because you put all that water into the ice caps. Then you can pretty much walk anywhere on the earth. Once the ice caps melt, a lot of these animals that were driven, non-predatory animals that were driven to different places in the earth, they'd be trapped to where they are. But the carnivory wouldn't be the same as it is today then, especially if a lot of the animals were still used to a vegetarian diet. A lot of the habits of carnivores today would have occurred sometime after the flood through epigenetic means, epigenetic switches that can be turned on when a lot of these non-carnivores become carnivores. So I think we're looking at animals today, what they eat today, and assuming that's exactly how it was in the pre-flood world and in the immediate post-flood world. So I don't know. I yield there.
All right. Next question for Jackson. In the book Phosphate Rocks of Carolina, it was stated unequivocally that human bones and arrows were found buried in the same rock layer as dinosaurs. Jackson, how do you refute? If you've even heard of the book, of course.
I've never heard of this idea, so I don't know how to refute it.
Okay. Have you, Donny, have you heard of
Can you repeat the question?
Yeah, it's about the phosphate beds of South Carolina. In the book Phosphate Rocks of Carolina, it states that human bones found with dinosaur.
Right. Yeah, that's a good argument too. So there are some anomalous examples like the Ashley phosphate bed where you have human bones mixed with dinosaur bones. Okay, so we have examples of that, which I believe the evolutionists would say like there's maybe some overthrusting going on. Of course, they've got a rescue device, but again, when it comes to ecological zonation, and I'll screen share here real quick just for the audience, whether or not we found humans and dinosaurs together. I mean, it's amazing that it seems like that's what we find in the Ashley phosphate bed. When it comes to ecological zonation, we wouldn't necessarily expect have humans and this is what I wanted to go to. Humans and dinosaurs living in the same areas. In Genesis chapter 2, this is kind of what I hinted at earlier. The river coming out of the Garden of Eden is described as dividing into 4 rivers, which may imply the Garden of Eden, with its fruit trees and other angiosperms, mammals, and man, was at a high point geographically. The rivers flowing downhill to the lowland, swampy plains bordering the shorelines where the gymnosperms grew and the dinosaurs lived. This would explain why we don't find human and dinosaur fossils remain together in the geological record. Dinosaurs and gymnosperms only fossilize together, and angiosperms only fossilize with mammals, and man higher in the record, separate from the dinosaurs and gymnosperms. This is the same thing with flyers too, like pterodons, pterodactyls, birds, bats, different ecosystems. Just because they fly doesn't mean that we'd expect them to all live in the same ecosystem. Same thing with the ocean, sharks, whales, so on and so forth. But I think the Ashley phosphate bed example is bonus on top of the creation model. We wouldn't necessarily expect it or predict it because of ecological zonation, but the fact that we do find them together is incredibly contradictory to the evolutionary based model and the geological column. We can give Jackson the last word if he wants.
Yeah, I don't really get the ecologic zonation thing, because it's assuming that everything's buried immediately and stays there. In a turbulent global flood that lasts a year, I would think things would get mixed around pretty badly. That's all I have to say.
All right. We're going up on about 2.5 hours, so I'll blow through some of the questions. Norman Bates wanted to know, do you think humans could ever evolve into leprechauns?
No.
It would be pretty cool if they could. If they had a pot of gold. Of course, that would devalue gold. So, but no, I don't think so.
I think we see humans today accumulating 100 new mutations per person per generation. So it's funny. You could make a ton of funny questions, derive a lot of funny questions based on the evolutionary model, since according to the evolutionists, humans came from non-human-like ancestors, which ultimately go back to a single-celled like ancestor billions of years ago. So, I mean, are there really limits? Maybe they'll say there's physical limits. For example, can we get a dog the size of a whale or a dog the size of a flea? I mean, there's animals that exist that are the size of a whale and the size of a flea. They're called whales and fleas, but to take a single-celled like ancestor into a whale, why are there limits according to evolution? But we know, based on the empirical data, humans are accumulating deleterious mutations. They're not evolving into anything other than humans. As a matter of fact, there's a shelf life on humans, which is why our hope is in Christ. And that's why we want Jackson today to believe on the Lord Jesus Christ, because genetic entropy is a reality. There's been no rebuttal. All the best rebuttals have been systematically analyzed and falsified. And our hope is in Christ, and our hope is in heaven. So I yield.
Well, I don't think that genetic entropy and Christ are, necessarily needed together. But I think genetic entropy is not the greatest idea. I mean, I've been doing a lot of research on that myself, and I'll provide the numbers later, I guess. But if viruses with high mutation rates aren't going through some kind of genetic entropy, I don't see how we're going through it in 6,000 years from the patriarchs to now.
Are viruses alive?
They're made of genetic material. So, I mean, Sanford himself used H1N1 as an example for genetic entropy. So
now here's the thing. We talked about viruses a lot during the discussion. If viruses are created DNA elements, they started in the host rather than predated the host. Okay. But genetic entropy is written for longer lived animals, for example, which we know have higher mutation rates. Why would we then look to viruses, which exist in us, and then viral like elements as well, to refute the concept of genetic entropy?
I don't know. They're just made of genetic material, and they're replicating and they're mutating very quickly. I think it would apply.
We've done thousands and thousands and thousands of experiments, numerical simulations, all realistic parameters, different types of selection, different types of amounts of beneficial mutations, different population sizes. And what we always get, and these are documented, is degeneration. Selection can only ever slow down the process and not stop it or solve it. And what they've put forth in terms of truncation selection, synergistic epistasis, mutation count mechanism, they've been looked at, documented, analyzed. You can read papers on it and articles on it. They've been falsified, but you want to look to viruses. Doesn't really make much sense. It sounds like you just don't want to believe it. And I'm not trying to be rude. It just sounds to me like how come all of these evolutionary population geneticists like Lynch, Kondrashov, Kimura, how come they never say, well, there's no point in, in looking into the mutation accumulation problem because, wow, viruses exist. How come we don't see them saying that? I mean, they recognize it's a real problem. Kondrashov said, why are we not extinct or dead 100 times over? How do you respond to that?
Yeah, he did say that. I forget why he said that. I read part of that. Yeah, I mean, I don't have a great response to you. Again, just to say that viruses are genetic material, and I think they're a good analog.
It's okay. We can use that for the after show. If you're going, that is.
Yeah, I'm going.
All right. Thanks,
the
John.
Continues all night.
Yeah, thanks for the super chat, John. Just a reminder, that's where that's going to be right now as we wrap this up. So, Donny, if you wanted to let everybody know about future debates that are coming up, and you can close it down.
John's like, this is the Q&A, not a further debate. Yeah, this has been a long one, a detailed one, a technical. I'm exhausted. Jackson, I'm sure you are too. I'll probably get a sandwich in, or I don't know, another coffee or something. This was a technical, fun debate. Lots of good subjects and topics discussed. Good audience Q&A. The next debate that we have is Jackson Roe. You better get some rest, my good man.
Yeah, I need to do some more research on that. Also,
we'll try to not make
some slides.
We'll try to not make it. So that's going to be fun. You and Professor McQueen. And then right after that, the big round 2 between David Neff and Dr. Dino. And all I can say is go have a look at the upcoming live stream section because we got about 40 more debates. So, Matt, great job moderating. I appreciate it.
Except for the end there when it turned into a second debate.
Matt, you're on mute, brother. Whoops. I'm just saying, good job. Couldn't even get through it. Thanks.
You did a fantastic job. There we go. 2.5 hours, man. It didn't feel like that though, Jackson. Tons of great points discussed. I hope the audience had a good time. Looks like we still have 90 people. So, we kept everybody's attention. Thank you for preparing and bringing some good arguments that we can discuss, Jackson. So, guys, we'll see you over at the after show. Logical, plausible, as Matt calls it, possible.
I can't remember the order
plausible, probables channel. We'll see you there for another epic after show. I'll probably join in about 45 minutes. I got to get some food and take a little break here. So,
and some coffee.
Yeah, guys, God bless. Thanks for tuning in. We'll see you in a couple days. Standing for Truth is out.